Right. As I mentioned previously, I'm not going to emphasize too much on the following uh, sections. You know, this allele frequencies in populations and the molecular clock and so on. But I'm just going to briefly go through it. Right. So if this one talks to you about, okay, when you have mutations, if it is a neutral mutation or it is not so disadvantageous, then you will see the mutations quite a lot of time in a population, and these are calculated in terms of frequencies. The frequency of alleles, right? For example, you have the ABO blood group. You know, you have the A allele, B allele, and the O allele, and so on. So there's not much. There are variations, but it doesn't have any effect on the fitness. So that's why you can see this in the population. So the frequencies in a given population, it depends very much on mating patterns, mutation rates, migration, natural selection, random genetic drift, and gene conversion. So these are, again, principles of population and genetics. You're talking about how the mutation behaves in a population after several generations. You know, what happens to it. So terms like, you know, if you see further, whether it's a negative selection, we're talking about selection pressure. And when if there's a selection pressure, that means the mutation is if there's selection pressure, that means the mutation makes the individuals less fit. So that means if it's less fit, that means it's not going to survive. And because it's not going to survive, so the survivability of the mutation is going to be less as well. Yeah? If the mutation gives you additional advantage, so that means it's going to survive. So the frequency, you expect to see more and more of that mutation in the population. So this is what is basically talking about. And of course, this changes if the population is large. Then you will not probably see this effect too much. But if the population is small and then there are bottlenecks, and bottlenecks means events or situations where gene flow has been disrupted. I mean, for example, you have a population and then there's a big flood, and because of that, the population is now divided into two subpopulations with maybe a stream in the middle. So now they are not intermixing. And because of that, if one population is too small, then it may not be able to survive, and then the frequencies will change, right? Because it's now behaving in a smaller subset than the larger population. So these are the things that can change frequency of alleles. And then what about this molecular clock? Okay, now let's look at two different mutations. One is synonymous, the other one is non-synonymous. Synonymous means it's the same, so that means there's no effect. All right. Non-synonymous, let's say it is deleterious. That means it gives you a negative effect. If you look at the population, which ones would you see more frequently, the synonymous one or the non-synonymous one? I would expect the synonymous one to see more frequently, because again, you don't see any effect. So that's why the mutation can occur without any hindrance. But if there is a mutation, that the non-synonymous mutation may, in most cases, we're talking about negative effects. So you'll see it much less. That's why diseases are much less, right? So if you compare two genes, right, two proteins here, right? Here you look at this. The rate of non-synonymous substitution for an evolving protein is indicative of the intensity of the selective pressure to maintain its structure. What it's trying to say is that if the protein, sometimes the protein can allow changes in its amino acids, but it is not affecting its function. Yeah. So that means it's not so strict. But another protein may the slightest change will result in non-functionality. So if you compare these two proteins, this one is more is more likely to evolve because it can allow changes, correct? But this one cannot. So that's why I'm saying the rate of non-synonymous substitution for an evolving protein is indicative of the intensity of selective pressure. So that means if it's rapidly evolving, that means there's not much pressure on it. The next sentence, the rate of synonymous substitution for histone and albumin proteins are about the same. Right? The rate of synonymous. So that means there's no effect. So of course, these two proteins, histone and albumin, the synonymous mutation rate is the same. However, although the non-synonymous substitution rate for albumin is several hundredfold greater than that of histones. What is trying to say? It's saying that albumin is less strict. It allows for a lot of non-synonymous substitution, whereas histones are very, very strict. Yeah, if you have changes, then it probably will not survive. So that means it dies off, so you won't probably see it. So through evolution, you will see that albumin has got many, many different types, but histones will not be that variable. So this is what it's trying to say. And the study of this rate of change between these proteins, this is what this is what we're talking about, the molecular clock, right? So this has given rise to the concept of molecular clock, where it talks about the measurement of evolutionary time as the rate of neutral evolution. It's not something easy to understand, but it's just trying to say how fast the gene is evolving, and this is indicative of the organism itself, right? So the molecular clock is not constant because it's depending on the gene, as I mentioned. You know, albumin and histones, one is very strict, the other is not. Different genes vary in neutral substitution rate as well as their amino acid replacement rate. So while earlier we said, you know, for histone and albumin they're the same, but some genes, although some genes show differences in synonymous. Why? Because of the sequence content. Remember, Cs are more prone to mutations previously. So that means if there is a gene that has more Cs compared to the other, although they're synonymous, but you'll find that the other one is more variable, right? So it's a bit complex concept, but this is what they study. They study the rate of evolution of genes and proteins and come up with a molecular clock. See you later.